affects inflammation, acute phase reaction, immunoglobulin synthesis, and the activate states of B cells and T cells. Interleukin-6 stimulates B cells to proliferate and differentiate into plasma cells and induce CD4 T cells to produce greater quantities of both pro- and anti-inflammatory cytokines. Only one interleukin-6 receptor has been identified, and it consists of interleukin-6 Rx, or the interleukin-6 specific receptor, and GP130, the common signal transducing receptor subunit utilized by the several cytokines. Binding of interleukin-6 to the interleukin-6 specific receptor induces dimerization of GP130 with the alpha subunit. Homodimerization following interleukin-6 binding caused conformational changes in GP130 that expose tyrosine residues in the intracellular portion of the molecule. Through a series of phosphorylation reaction, genes for acute phase proteins such as CRP, C3, and fibrinogens are activated, as is interference regulatory factor 1 (IRF). B and T cell genes are turned on the same manner. Now we're going to talk about the chemokines. Chemokines are a family of cytokines that enhance motility and promote migration of many types of white blood cell toward the source of the chemokines, or chemotaxis. Most of the chemotactic activity of leukocytes is regulated by the activities of chemokines, including the response to infectious disease, autoimmune inflammation, cancer, and the homing of lymphocytes to all lymphoid tissues. The chemokines are classified into 4 families based on the position of N-terminal cytine residues. The first group, the alpha or CXC chemokines, contains a single amino acid between the first and second cysteines. The second group, or the beta or CC chemokines, has adjacent cysteine residue. The third group, the chemokines, lacks one of the cysteines. The CX3C, the last major group, has 3 amino acids between the cysteine. Chemokines play a key role in the initiation and development of inflammatory response in numerous disease processes. Currently, over 40 chemokines and 20 chemokines receptors have been identified. Both tumor necrosis factor alpha and interleukin-6 are among the many cytokines that induce chemokines production in the inflammatory response, combined with cell adhesion molecule. The chemokines facilitates the intravasation of leukocytes into the tissue. Leukocytes rolling on capillary endothelial cells activates their chemokines receptor in the presence of chemokines. This, in turn, activates integrins or cell adhesion molecule on leukocytes and lead to firm adhesion to the endothelial cell. Shared expression of chemokines receptors among different types of leukocytes allows for the co-localization of multiple cell types to the damaged tissue and help to broaden the response to tissue damage. The gradient of chemokines concentration enables the leukocytes to migrate between the endothelial cell into the tissue in the direction of increasing chemokines concentration. The spectrum of chemokines and cytokines expressed in the inflammatory response determines the type of cell that responds and the gene that are turned on in response to the stimuli. The type of cell surface receptor expressed by leukocytes are often developmentally regulated. For example, immature T cells possesses only one chemokines receptor related to lymphoid tissue homing. Only mature T cells express the receptors that allow them to participate in an ongoing immune reaction. The chemokines receptors CXCR4 and CCR5 are utilized by HIV as co-receptor for infection of CD4 T lymphocytes and macrophages. Individuals with certain polymorphism in this chemokines receptor are long-term non-progressor. They remain asymptomatic, have normal CD4 T cells, count and normal immune function and have low or undetectable viral loads. The altered protein sequence of the receptor blocks or diminishes the virus' ability to enter the cells and thereby increase the infected individual chances of survival. The CCR5 triangle 32 or polymorphism is a 32BP deletion in the CCR5 gene and is the most important of the host-resistant factor. Homozygous individuals are protected from HIV infection while heterozygous person exhibits longer period from infection to AIDS development. In addition, certain polymorphism, CDF1, the ligand for CXCR4, and RANTES, the ligand for CCR5, can block the virus's ability to bind to and enter T cell and can delay the progression to full-blown AIDS. Now, let's talk about the tumor growth factor beta. The transforming growth factor beta. The transforming growth factor beta superfamily is composed of three isoforms: tumor growth factor beta 1, beta 2, and beta 3. Tumor growth factor beta was originally characterized as a factor that induced growth arrest in tumor cells. Later, it was identified as a factor that induced anti-proliferative activity in a wide variety of cell types. Activity TGF-B beta is primarily a regulator of cell growth, differentiation, apoptosis, migration, and the inflammatory response. Thus, it acts as a control to help downregulate the inflammatory response when no longer needed. In the immune response, transforming growth factor beta function as both an activator and an inhibitor of proliferation depending on the developmental stage of the affected cells. Transforming growth factor beta regulates the expression of CD8 in CD4 and CD8 thymocytes and acts as an autocrine inhibitory factor for immature thymocytes. It inhibits the activation of macrophages and the growth of many different somatic cell types and functions as an anti-inflammatory factor for mature T cells. Transforming growth factor beta blocks the production of interleukin-12 and strongly inhibits the induction of interferons gamma. In addition, the production of transforming growth factor beta by T helper cells is now recognized as an important factor in the establishment of oral tolerance to bacteria, normally found in the mouth. In activated B cells, transforming growth factor beta typically inhibits proliferation and may function as an autocrine regulator to limit the expansion of activated cells. Now let's talk about the interferons alpha and beta. Interferons were originally so named because they interfere with viral replication. However, it is the type 1 interferons consisting of interferons alpha and beta that functions primarily in this manner. These interferons are produced by dendritic cells and induce production of protein and pathway that directly interferes with viral replication and cell division. In most cases, this helps limit the infection to one relatively small area of the body. Type 1 interferons activates natural killer cells and enhance the expression of MHC class I protein, thus increasing the recognition and killing of virus-infected cells. The type 1 interferons are also activates against certain malignancies and other inflammatory processes. For instance, interferons beta is efficacious in treating multiple sclerosis, although the exact mechanism of action remains unclear. Interferons alpha has been used to treat hepatitis C and Kaposi sarcoma, as well as certain leukemias and lymphomas. Now we're going to discuss the cytokines in the adaptive immune response. So cytokines involved in the innate immune response are produced by many different cell types and function mainly to increase acute phase reactant and to recruit white cells to the area of infection. In contrast, cytokines involved in the adaptive immune response are primarily secreted by T cells, especially T helper cells or Ts, and affected T and B cell functions more directly. There are three main subclasses of Th cells: Th1, Th2, and Treg, or the T regulatory cells. Each has a specific function and produces a different set of cytokines. Once the T cell receptor capture antigens, clonal expansion of those particular CD4 T helper cells occurs. Differentiation into Th1 and Th2 or Treg cell lineage is influenced by the spectrum of cytokines expression in the initial response. The Th1 lineage is driven by the expression of interleukins 12 by dendritic cells and is primarily responsible for cell-mediated immunity, while Th2 drives antibody-mediated immunity and are developmentally regulated by interleukins 4. T regular cells are derived from interleukin 10, responsive, naive T cells, and help to regulate the activities of Th1 and Th2 cells. Now let's go with the Th1 cytokines. The dendritic cell in damaged tissue produce interleukins 12 in response to certain stimuli such as mycobacteria, intracellular bacteria, and viruses. It is also produced by macrophages and B cells and has multiple effects on both T cells and natural killer cells. Interleukins 12 binds to its receptor on naive T cells and cause the expression of a new set of genes, including those that determines maturation into the Th1 lineage. Activation of Th1 cell induces high-level expression of interferons gamma. Interferons 12 also increase the cytolytic ability of natural killer cells. Therefore, it serves as an important link between the innate and adaptive immune response by enhancing defense against intracellular pathogens. Interferons gamma is the principal. We're going to talk about the interferons gamma. It is the principal molecule produced by Th1 cell and is affect the RNA expression level of more than 200 genes. Genes involved in regulation and activation of CD4 plus Th1 cells, CD8 cytotoxic lymphocytes, natural killer cells, bactericidal activities, interleukins 12 receptor, and interleukins 18 receptors are all regulated by interferons gamma. In addition, interferons gamma stimulates antigen presentation by MHC1 and MHC2 molecules. The increased expression of MHC class 1 and 2 molecules on antigen-presenting cell increases the likelihood of antigen capture and the involvement of additional lymphocytes. In addition to the above listed action, interferons gamma is a strong stimulator of macrophage and boosts their tumoricidal activity. Thus, interferons gamma enhance the immune response in a number of key ways. Interferons gamma production can be stimulated in mature Th1 cells by two means: ligation of the T cell receptor by MHC peptide antigen presentation, or cytokine stimulation by interleukins 12 and interleukins 18. Interleukins 12 and 18 act synergistically to stimulate interferons gamma production, even in the absence of TCR ligation. In addition, interleukins 12 and interleukins 18 can activate interleukin interferon secretion by CD8 lymphocytes. Interleukins 2. Th1 cells also secretes interleukins 2 in addition to interferons gamma. Interleukins 2 is also known as the T cell growth factor. It drives the growth and differentiation of both T and B cells and induces lymphatic activity in natural killer cells. Interleukins 2 and interferons gamma induce the development of Th1 cells, which in turn induce macrophages activation and delayed type